So, I focus on hoofed mammals, primarily the ungulates. And ungulate actually means that they walk on their tiptoes, or their ungulophalanges, the last bits of the toes. And particularly, I focus on artiodactyls, which are the even-toed hoofed mammals. And these are all members of the Protoceratidae, or the Protoceratids, and this is an extinct lineage of artiodactyls that is exclusively North American. And I'm going to talk to you about why they've been such a problem and why they're so interesting. So, Protoceratids have been a problem since they were first described. And they are very clearly members of the even-toed hoofed mammal radiation, the artiodactyls. And these are all members of the artiodactyls. So Artiodactyla is a very large group of animals. It includes all of these terrestrial mammals and a few marine ones, as it turns out. So it includes cattle, it includes giraffes, it includes camels, it includes hippos, it includes pigs and peccaries. It's a very diverse group. It's the only really diverse group of large mammals that is still alive today on the planet. So, sorry. That radiation is part of a group of animals called the Laurasia. So this is the phylogeny, or the family tree of mammals. Okay. And up here at the top, you can see the monotremes and marsupials are among the earliest diverging lineages of mammals, and they diverged away from the rest of the mammals, who we call placental mammals, right? And so all of these mammals belong to the Placentalia. And here you can see this radiation is called the Afrotheria. That means African beasts. And they are all things that today still inhabit Africa, primarily. And then we have the Xenarthra. So an anteater is a Xenarthran. The Urocontoglires is us, the primates, the rabbits and rodents. And then this Laurasia includes things like hedgehogs, carnivores, pangolins, horses, the perissodactyls, the artiodactyls, and bats. Okay, so that's where we're talking about within the mammal family tree. And within the artiodactyls, we have four main modern lineages that we talk about. So we have the pigs and peccaries, the Suina, the hippos, the Hippopotamidae, the Tylopoda, which today is represented by the camels and the llamas, and the Ruminantia, which is everybody else. So cattle, sheep, goats, muskox, deer, it's antelope. It's a very large radiation. Okay, and this is the traditional family tree that was used when I was a student, that most of you may have seen, where it's thought that camels and ruminants are closely related for a number of reasons. They have a different body form from pigs and hippos. They have fewer toes, they have longer legs, and they gut ferment. They chew their cud, right? This was all upended while I was in graduate school. When it became obvious from molecular data from the genes that whales are closely related to hippos, that messed everybody up for a long time. And then subsequently, new fossils were discovered in Egypt of early whales, Egypt and Pakistan, that showed that the earliest whales actually have ankle joints, just like artiodactyls. And they do belong really clearly up here within the Artiodactyla. So this is the molecular phylogeny, and that is basically what we are operating within now, because it's become very clear that Artiodactyls have a lot of convergent evolution. They like to keep doing the same things over and over and over and over again in response to different climatic conditions. So as of today, there's some argument about whether camels are more invasive than the pig lineage, but essentially camels are not closely related to ruminants. Instead, all the things they share with ruminants seem to be really convergent, right? So they're long-legged, they have reduced their side toes, and they gut ferment, and they acquired these things independently. from ruminants. Okay? So that's the backdrop. Now, protoceratids have been known for a really long time. The first ones were actually described by O.C. Marsh, Othniel Marsh of dinosaur fame. Right? They're well known from the North American fossil record. And they are exclusively North American. This is a fairly derived form called Synthetoceras tricornatus. And you can see why it's called tricornatus. That means 3 horns, right? And it's got 2 orbital horns and a nasal horn that is actually split, okay? This is a derived feature in this lineage. The earliest ones are small and don't have any horns. But as soon as they get large, we start to see horns showing up. And I say horns, they're not actually horns in the way bovid horns are formed. And we will get to that in a little bit. Well, actually right here. So there is independent evolution of what we call headgear in the artiodactyls. They do this a lot, right? You're aware, probably, well, certainly anybody who hunts is aware, that horns, such as we see in antelope and cattle and so on, are not the same as antlers, right? Horns are permanent. They have a bony horn core and a keratin sheath. And the keratin sheath lasts all life. It grows throughout the individual's lifetime. Whereas antlers regrow every year. They are covered in skin, and then the skin dies back during the rutting season and falls off. And at the end of the mating season, the antlered base is resorbed and they drop off. So these are a significant investment on an annual basis for male cervix, for male deer, right? In addition, you have ossicones that we see in giraffe. These form completely differently. They form as little ossifications that float under the skin and eventually fuse onto the skull, and they're always skin covered. They are used somewhat in male-male combat in giraffes, but the main weapon that giraffes use is the rest of the head. They actually are whacking each other in the neck with their heads. Giraffe's neck fight, okay? And fossil giraffes, there's a whole range of really amazing headgear. There's one that looks like a ceratopsian. Like I have accidentally turned around in the museum and thought, is that a ceratopsian? Oh no, it's a Shibatherium. It's really confusing. And then we have pronghorns. And pronghorns have this interesting sort of hybrid morphology. They have a permanent bony core and a keratin sheath like a bovid, but they shed the keratin sheath every year just for fun. So we have a lot of convergent evolution going on in artiodactyls with headgear. And it turns out that these guys are the original ones with headgear. We don't have a very good understanding of how the headgear grew. Nobody has yet studied it. I keep hoping I'll be able to convince a student to take on a project of doing some histology. So sectioning these in different life stages to try and figure out how they were growing. But they do seem to be growing out of the frontal bone rather than growing and attaching on. We don't have any fossil specimens that show any kind of suture at the base of the horns. Okay. This is the last form in the record. This is Cyptocerus, and this one is from Florida. And we have protoceratids appear in the record in North America from the Middle Eocene, so the Uinta land mammal age, for those of you who know about those, all the way to the Pliocene. And as far north as Saskatchewan, and as far south as Panama. Okay. So I wanted to show you this because there is somebody actually working for a change on an animated feature about fossil mammals, which is very exciting. And this is a bit of the trailer for this. And I wanted to show it to you because they've reconstructed a protoceratid. There is the protoceratid. That is Synthetoceras tricornatus. Okay, so, and those are some camels. Oh, come on. There we go. Okay, so here is the distribution in the fossil record. You can see all the way down to Panama. And the Panamanian material is some of the latest material. The youngest protoceratids in the record are actually Canadian, or the oldest rather. They are from Alberta, not Alberta, Saskatchewan, sorry. They're from the Swift Current Creek fauna in the Cypress Hills of Saskatchewan. And they are well represented there. We have a number of, for those of you who've sorted the Swift Current Creek material, we have a number of teeth. We don't have much else, as those of you know, that's a really heavy duty conglomeratic fluvial deposit, and you don't get a whole lot of whole material out of it. So whole teeth is about the best we can do. But we have them from Swift Current Creek. We also have them later in the Cypress Hills Formation in Saskatchewan, in the Chadronian. They're also common in the White River Group and the Chadron Formation. So late Chadronian and early Arellan, so about 34 million years ago. The oldest material is a little over 40 million years ago. And they persisted all the way up past actually the Clarendonian into the early Pliocene. So they're quite persistent. And as I said, exclusively North American. And we have quite a lot of good material. Unlike the Cypress Hills material, we have some really beautiful articulated specimens. And so this is the diversity of the family with a human for scale. And what you can see is there are a couple of really tiny hornless forms. So the hornless forms from the Eocene, and these are known from Saskatchewan as well as localities in the US. And this one, and then we start, as soon as they get a little bit bigger, we start seeing headgear, right? And today, if we look at hoofed mammals, there are no hoofed mammals with headgear that seem to have, they basically seem to have evolved headgear as soon as they hit about 18 kilos. And below 18 kilos, we don't tend to see horns or antlers or any of those. We see enlarged canine teeth that the males use to fight with. So it's pretty clear from all the living forms, these are used by males in male-male territorial combat, and in the various forms, they only seem to get them around that 15 to 18 kilo range is when they start to evolve headgear. And they all evolve headgear in the same spots. It's always over the eyes and over the nose. There's not a whole variety of other locations on the head where you see headgear popping up in the hoofed mammals. Okay. So here we have, this is Leptoridon, which is the oldest one in the record, Heteromeryx. Then we have Protocerus and Paratocerus and Syndiocerus over here, Synthetocerus and Kyptocerus. This, for those of you who've met my grad student, Selena was working at the University of Nebraska State Museum, and we've got some pictures of them with protoceratids. They were studying them because of this phylogenetic problem of who are protoceratids related to. And this is a beautiful specimen, complete, including in there a fetus preserved. This is from ashfall fossil beds. And we get amazing preservation. That is an ashfall site. And so we get pregnant protoceratids, pregnant oreodonts, pregnant horses, pregnant rhinos. There's a number of pregnant specimens from this site. So it's an amazing site. Now this is a reconstruction of the late Oligocene and early Miocene faunas of North America. So this is actually based on some of the sites in Nebraska, and you can see here a whole variety of mammals. This is my plug for how cool fossil mammals are, not just protoceratids. Those are calicotheres. They're related to horses and rhinos. Over here, you can see some oreodonts, early horses. There's our protoceratid. Okay. And over here, a whole host of different forms of camelids, of camels. And yeah, 5 minutes. Oh my goodness. Speed this up. Okay, so here are some more shots of these guys, and later on, and through time, we get bigger and more funky horns. Okay. These are the specimens. They're quite spectacular. And we've been doing some work to try and figure out who they're related to, because traditionally they were grouped with the ruminants And then subsequently, other people argued they were closely related to camels. And the camel idea makes sense because camels, until 5 million years ago, were also exclusively North American. But it's also possible that they're more basal forms. And in the newer interpretation, you can see that being related to ruminants and being related to camels is quite a bit different proposition. And then there's all these other basal positions that they could fit into because of the problem of convergence. So in the 90s, some colleagues did some early CT scanning. These are llamas. And this is showing you the ear region. And I want you to focus on, this is the ear, the inside part of your ear in your skull, in a bone called the petrosal. And I want you to look at the shape here. Notice it's kind of flat. And then there's a big space here. And that big space actually houses a lobule of your brain called the paraflocculus. We compare that. This is a protoceratid. Nobody had ever seen the inside of a protoceratid skull. This is one of the first CT scans used in research in paleontology. And you can see here that in the protoceratid, they have a big wedge here between where the cerebrum of the brain would be and the cerebellum would be, right? There's a big ridge. We go back, do you see a big ridge here in a camel? This was one of the first hints that the camel idea might not hold up. So we did some more work. Here's a camel, no ridge. Here's a ruminant with a nice ridge. This is Protocerus, so this is another more basal protoceratid we looked at to see if it was the same as Syndioceras. And lo and behold, it also looks more like a ruminant. So we did some more work on looking at the ear in more detail. So these are the protoceratid skulls. And I should point out that here you can see this one is the male and it has horns, it has some protuberances, and this one is the female and it does not. And this is one of the best examples of sexual dimorphism in the fossil record. We find these in the same horizon of the same deposits. So it's very clear that these are sexually dimorphic populations. So this was a confusing problem because somebody else at the American Museum, Maureen O'Leary, published what she said was a petrosal bone from Protocerus at the same time as we had just scanned it in the skull. And this petrosal doesn't look anything like the petrosal that we rendered out of the skull. And this was an isolated specimen. So we think that the specimen she described, which was an isolated specimen, had been wrongly associated with the genus Protocerus and is actually probably a camel, because it looks more like a camel. And then there was this argument that camels and protoceratids might be related, in addition to the other features which everybody had mentioned, which are the long legs and the shape of the head. There was a feature in the neck that people were talking about. This is a neck vertebra. This is a bison on top. And this is a llama on the bottom. And you see this hole here? That's where your arteries, your vertebral arteries, penetrate your neck vertebrae, right? Bringing blood to your head. And in all of the ruminants, they show the ancestral mammalian condition where it's a nice hole straight through. Can you see a hole that's going straight through the side in the camel? It actually comes out inside the vertebral canal, the neural canal. It's a weird pattern that you see in camels. And in the literature, it was reported that protoceratids had the same thing. Those of you who know Selena will know Selena doesn't take anything on faith. And so Selena went to look while they were there at the protoceratid neck vertebrae. Guess what? Both the primitive and the more derived protoceratids don't show anything like a camel neck vertebra. They show the ancestral condition. So the ear region looks more like ruminants than it does like camels. And the other features that it's supposed to share with camels are based on what is clearly a petrosal that doesn't belong to a protoceratid. The neck vertebrae don't look like camels. But most of the same features we see in the neck for Protoceratids are the ancestral conditions, so they don't actually tell us who they're related to, they just tell us they're not camels, right? So we have a whole bunch of features that are kind of ancestral. Does that make these ruminants? Probably not. And they're not ruminants for a couple of reasons. One of which is that they lack some big features that ruminants have. So all living ruminants, so cattle, sheep, giraffe, all have a fusion in their ankle of some bones called the navicular and the cuboid. And they're not fused in Protoceratids. They have the ancestral condition. And so it's most likely that the Protoceratids belong somewhere in the stem, either as stem ruminants or stem camels or even more basal. But we need a lot more work on these guys to really understand who they're most closely related to. There is a tiny hint from Selena's work on ears right now that these might actually be related to oreodonts, but that needs a lot more work. Anyway, I'm happy to answer any questions you might have about these weirdos. Yeah.
I was just wondering if you might like to make any comments on the extinction of some of these species or.
So these species are leaf-eating mammals. They're not grazers. They don't eat grass. They didn't eat grass. We have no evidence that they ever consumed grass. And they are never very common compared to, like, we find camels everywhere. These are always rare elements. And so it's quite likely that the vegetation and types of ecosystems they were living in just vanished. They're not very common late in the record in the Pliocene. And so I think the ecosystems petered out as grasses took over and the continent dried.
So there's no possibility that the size of their headgear became so massive that.
No, it scales pretty nicely with their body mass. That's also true for all the deer. So you hear that story in sometimes that the Irish elk went extinct because their headgear was so big. If you look at the size of the skeleton, it scales perfectly the same as it does in moose and elk. Their headgear isn't any bigger for their mass. They're just really big deer. Yeah.
Earlier you said that there was a sort of ceratid that looked exactly like a ceratopsia. What was that?
Oh, it's actually a giraffid. It's a fossil giraffid. It's called Shivatherium, Sivatherium. And it has all these funky horns all around its head. There's a whole bunch of really interesting fossil giraffids in Asia.
Thank you.
Yeah.
When reconstructing the life patterns of Protoceratid, is it a bad idea to assume that it's probably just skin covered for the ornaments, or?
No, I think that's the most likely thing, that they're growing right out of the frontal bone and that they were skin covered. We have a good example of, giraffids have that kind of structure, and it's pretty clear these are never shed, unlike some of the fossil pronghorns, and certainly the cervids. But it'd be really good. We have enough material that we should be able to do some histology on them by sectioning them, if we can get permission. Yeah.
Why would only the larger species have headgear?
So it's a really good question, and I don't think we have a good handle on why that's true even in modern ungulates. But if you look at the smaller deer, like musk deer, they have big teeth and don't actually have much in the way of antlers. So this is true across the board. And there are size reductions in lineages with antlers and horns that they retain them, but the ancestral forms, like mouse deer, they just have fangs. It's presumably a question of the ecosystems that they're moving through because of their size. Yeah.
Some of the skulls in your photographs, they chew that fangs.
Yeah, they retain the fangs. They just add headgear.
Oh, okay. Why not?
It's fun. Yeah. It's harder to infer that without bone beds to estimate, how many individuals were living together. But presumably not completely dissimilar from what we see in artiodactyls today that have headgear, right? So think about, there's a variety of different strategies in ungulates. You can see small territories, you can see herd behavior. It just, there's a bunch of factors that play into it. So it's harder to assess without a spectacular deposit with one population preserved. Yeah.
The question of finding lots of the well-preserved species that were pregnant.
Yeah.
Does that give you an idea of their seasonal cycles, or like canoes, they all give birth at once?
So there's, as far as I know, there's only one pregnant protoceratid in that assemblage. What it does tell you is how many animals were around that watering hole when the volcano went. Because there's quite a diversity of organisms preserved there at that one horizon. Yeah.
On the image there where you have the different species being found, A, B, C, D, how it kind of goes down the North American margin. Is that like a migration
It's
pattern? or is
no,
it like-
because that's the whole fossil record of them. So that's actually like through time and different lineages of them. So I think that's more reflective of the kinds of ecosystems they lived in.
So climate change, environmental changes kind of
Yeah.
pushes them down and then potentially pushes them back up.
Yeah. Yeah, and as I say, they're not as common as camels, so they clearly lived in much more restrictive environments, whereas camels seem to have been very resistant to drying out, and we see them all over until 11,000 years ago when they went extinct here. Yeah, Christina.
I was just wondering, well, you show that artiodactyls have such a variety of different headgear.
Yeah.
When do you think we will see that in artiodactyls?
I think for whatever reason, there is some genetic regulatory mechanism available in artiodactyls that is basically easy to turn on and off, and you don't have that same growth, that same developmental pattern available in parasidactyls. And the one group of, well, the two groups of horned parasidactyls I'm aware of only have nasal horns, and one of them, they're keratin, not bone at all, right? Yeah. So different genetic background, I think. And given how often certain patterns repeat in artiodactyls, it's pretty clear they keep doing the same things. Thanks.
Thank you very much.